Hi everyone. In this video, we're going to learn about fatty acid anabolism. Anabolism, when we talk about the term anabolism, we have to understand that it means synthesis, right? So, anything that is undergoing synthesis is called anabolism. And then the process of fatty acid synthesis or anabolism is also known as lipogenesis. The site of synthesis of the fats is the cytosol, that means in the cytoplasm, right? Now we have to be familiar with the process called glycolysis. You do not have to know all the steps, but you have to understand that um, you know glycolysis is when a glucose is broken down into pyruvate, right? So when a glucose is broken down after these many steps, it's broken down into pyruvate, then it is known as glycolysis. And pyruvate is that particular molecule compound which is going to further participate in the process of fatty acid synthesis. So glycolysis also takes place in the cytoplasm, right? So you just have to know that glucose, if you cannot remember all these steps, of course I'm sure you don't remember, but still, like, just remember that glucose has to, like, finally converts into pyruvate. So now let me show you, like, my diagram and try to explain what exactly is happening. So as I show out here, that the glucose is being converted into pyruvate, right? Which is a 3 carbon compound, and then the pyruvate, all these process is taking place in the cytoplasm. And then after pyruvate is being synthesized, pyruvate will go inside the mitochondria of the cell, and then a pyruvate, which is a 3 carbon compound, can undergo two processes. It can either undergo a decarboxylation means like removal of 1 carbon compound, and then after 3 carbon it can become a 2 carbon compound, and this 2 carbon compound is known as acetyl-coenzyme A. Now this pyruvate can undergo carboxylation, that means addition of one extra carbon, and then it can become oxaloacetic acid, which is a 4 carbon compound, right? Now, acetyl-coenzyme A and oxaloacetic acid can react with one another and become and synthesize citrate. This citrate has two paths. It can either undergo TCA cycle, or it can get outside of the mitochondria, and then after it comes out of the mitochondria, there is an enzyme which is called citrate lyase. Citrate lyase can break down citrate to the compounds which was before, that is the acetyl coenzyme A and the oxaloacetic acid, right? Now oxaloacetic acid can undergo a change, and then it can form malate, and malate can finally undergo a change it can undergo a change and it form pyruvate, right? So the enzyme is the malic enzyme. Now there are other enzymes as well which participates in this particular in this particular thing, but I've not mentioned all the enzymes. So this oxaloacetic acid is a 4 carbon compound, right? Malate is also a 4 carbon compound, but when malate is being converted into pyruvate, it is this malic enzyme will it will perform decarboxylation. So 1 carbon will be removed from the malate, and finally the pyruvate will be converted, and this can undergo the same process like the same pyruvate over here, right? So this is about the cycle of pyruvate. Now the main hero of this particular the main hero of this fatty acid synthesis is the acetyl coenzyme A. Now this acetyl coenzyme A will further undergo some certain changes, and it will finally produce fatty acids. So let us try to see what exactly does happen. So in the cytoplasm, the acetyl coenzyme A is present there, right? And it is a 2 carbon compound. Now there is an enzyme called acetyl coenzyme A carboxylase. This, as I told you, that carboxylation means, I'm just going to write it down. Carboxylase means like to add a carbon, right? So we have to add carbon, and decarboxylase means like it will remove one carbon, right? So as you can see over here, that in the cytoplasm, acetyl coenzyme A, when it undergoes carboxylation, then this particular enzyme will add a carbon to the acetyl coenzyme A. And then there are other certain factors as well which participates in this particular thing, which is the biotin. Biotin acts as a cofactor, and it is also known as vitamin B7, right? So now let us try to see what exactly happens. So when I add one extra carbon to acetyl coenzyme A, now what I'm gonna get? I'm gonna get malonyl coenzyme A, which is a 3-carbon compound, right? So what are the things that are being used in this particular reaction? Acetyl coenzyme A, which is a 2-carbon compound. Then we have the enzyme, right, which is acetyl coenzyme A carboxylase. And then we have a cofactor, which is biotin, which is also known as vitamin B7. Then we have ATP, right, and bicarbonate ions. So all these things are gonna produce a compound called malonyl coenzyme A, which is a 3-carbon compound, right? So now let us try to see how does they look like. So that acetyl coenzyme A looks like this, right? And then malonyl coenzyme A looks like this. So I've just given them number. You can call it 1, 2 as well. And then I've given here 1, 2, and 3, right? So 1 carbon, 2 carbon, so 2-carbon compound. 1 carbon, 2 carbon, 3 carbon, so 3-carbon compound, right? Now, malonyl CoA is a very important compound because it will act as a building block for the fatty acids. So, what exactly does this mean? We're gonna learn it with the process. So, let us go dive deeper. So, okay, so if you are able to understand this structure, there is a carbon, there is a ketone group, and then a methyl group, and then a thiol group, which is attached to a coenzyme A, right? And then out here we have an acid group, then we have a ketone group, and then we have a thiol and a sulfur group, right? So out here it's going to be like the carbon has not fulfilled its valency. So just make sure that you write down that carbon has 4 valency, right? So now let us try to understand the process of fatty acid synthesis. So there are there are 4 steps in this process. The first one is the condensation, then we have the reduction, then we have dehydration, then again we have reduction, right? So if I have to just write it down in short forms, when we talk about condensation, when we talk about condensation, we always see that a water molecule is removed, right? But in this condensation, carbon dioxide will be removed. That means 1 carbon will be removed. Okay, so I'm just gonna one CO2 removed. Okay, now when we talk about reduction, we all know that reduction means like to add to add hydrogen, right? If I have to be very like simple, then it's like to add hydrogen, right? So hydrogen can be added by NADPH, right? So NADPH will add donate hydrogen, and then when it donates the hydrogen, it changes into NADP positive, right? So then we have the other one, that is dehydration. So dehydration means removal of water, And then finally we again have reduction. So reduction will be NADPH, which will finally change to NADP plus, right? So these are the 4 steps. So if you want to remember, you can remember by the first word, that is CRDR, right? So now the main 2 heroes of this process is actually ACC enzyme. So if you remember, I talked about ACC enzyme, that is the acetyl coenzyme A carboxylase, and then we have another enzyme which is fatty acid synthase 1. So we have to be specific, that is 1, right? So if you see out here, this is the structure of fatty acid synthase. So fatty acid synthase consists of a cysteine group. This should not be double bond. Okay. So fatty acid synthase, this is the enzyme. So this enzyme actually has a cysteine group to one end and acyl carrying protein. This is acyl carrying protein in another end. Okay. So and then they have a thiol group, of course. Now what will happen is, so just remember that cysteine and then acyl carrying protein will be there. So now let us try to understand the process, where everything is made up, like the fatty acid is made up. So as you can see over here, we have the fatty acid synthase 1 over here. Now what will happen is, there is an enzyme which is known as acetyl transacylase, right? So acetyl transacylase, will help us to join this molecule. So what exactly is this molecule? If you remember, I made this molecule like in above, if you remember. So this is the acetyl coenzyme A, right? Because it has a ketone group, a methyl group, and a sulfur, and then a coenzyme A, right? So as you can see over here, that this is acetyl coenzyme A, and then it is coming to this reaction. So when it comes to this reaction, this part will get cleaved off. That means the sulfur and the coenzyme A of the acetyl coenzyme A cleaves off, and then it will join to the acyl carrying protein part of the fatty acid synthase, right? So I have done here double bond. Just let me just make single bond. Okay. So now as you can see over here, that the acetyl group has joined itself to the ACP protein, ACP FAS part, right? So as you can see over here, that this part is being joined over here, right? Now there is another enzyme which is known as acyl transferase. So now what will happen is, as you can see over here, that the acetyl group was being attached to the ACP, acyl carrying protein part, right? Now this particular acyl group will be transferred to the cysteine part. So if I just have to write it down, I'm just going to write it over here. So S will be gone, and then we'll have a ketone group, right, and a methyl group, right, a methyl group. Now this ACP will have its thiol group back, right? So as you can see over here, that this has been transferred to the cysteine SH. Now let us see what happens. Now there will be another hero that will be coming in, that is malonyl coenzyme A, which is the building blocks of this particular fatty acid. Now malonyl coenzyme A, with the help of malonyl transacylase, this enzyme, it's gonna come over here, and then it's gonna join to the ACP part. Okay, so this malonyl coenzyme A, it's gonna come and then it's gonna join to the ACP part of the FAS enzyme, right? The FAS enzyme is actually a very big enzyme. This enzyme is a very, it has large components and everything, but I don't want to go to that. I don't want to go. I don't want to delve deep into this FAS. I'm just gonna keep it simple. So malonyl coenzyme A will come, and then after it comes, it's gonna shed its coenzyme A and then the SH, and it's gonna join with the ACP SH. So if I just have to write down, I'm just going to write it over here. So cysteine has an S group, right? Then it has a ketone and a methyl group. Now when we talk about the malonyl joining to the ACP, then we have, what kind of structure do you think we'll have? C double bond O. Let me just make it with another color. Let me just take this. So C double bond O, right? And then we have, what do we have? C, then we have another C double bond O, right? And then we have right. So if you remember the structure of malonyl coenzyme A, it was something like this, right? If you remember, I'm just going to show you over here. I'm not going to go back. So as you can see over here, that this group will be cleaved off, and then this will be joined, right? So I have joined it from this side. So 1, 2, and then 3, and then 4, right? So there are 3 carbons, and then we have an acid group at the end. So this is the thing about malonyl coenzyme A. So as you can see over here, that I have joined these malonyl coenzyme A over here, right? Now what will happen is okay, so now there will be a reaction. Okay, so this cysteine, this particular cysteine, it was sent above. First it was attached to the ACP group, right? Now it has been sent to the cysteine group. Now this cysteine will come and then attack this particular bond. Okay, so we can see that this carbon will this carbon, and then this carbon will form bond, and then C double bond OOH, that is the carboxylic group, will be cleaved off, right? So I'm just going to show you with a pink one. So this will be cleaved off. This will be O negative. So this will be cleaved off, right? And then when this will be cleaved off, this part is going to come and join over here. So this is going to go away, and then this part is going to come and then join. So let us try to see how does it look like now. I think I've made an extra step over here just to show you. So as you can see over here, that this and this will be condensing together. The enzyme which will be used is known as acyl-malonyl ACP condensing enzyme, right? And this process is known as the condensation, which we talked about in the just like 2-3 minutes before. So in this process, what will happen is a carboxylic acid, right? It is moving out of the malonyl coenzyme, right? Sorry, malonyl group, not coenzyme A. Malonyl group, coenzyme A already left. So this is actually going out, right? So we can see that one of the carbon molecule, one of the carbon atom is actually leaving, right? So this acyl group is actually contributing 2 carbons. This will also contribute 2 carbons, right? And then this group will be like, it will be cleaved. So together they'll make 4 carbons, right? Because it'll be 1, 2, 3, 4. This will be gone, right? So now let us try to see how does it look like. So now after they are joined, this particular cysteine will get back to its thiol group, and then this particular place will be again, free, and then there is a possibility that it can bind with, other upcoming molecules or compounds which will be coming. As you focus over here, we can see that there are 4 carbons, right? So we can see that 1, 2, 3, 4, right? So this particular compound is known as beta ketobutyryl ACP, right? Now why do you think it's called beta? Because we are taking consideration of this particular what do you say, carbon, right? So this was beta, right? So we are actually taking consideration of this particular carbon, and then keto because it has a ketone, and then butyryl because it is a 4-carbon compound, and ACP because it is attached to the acyl carrying protein. Now what will happen is, as we finish the process of condensation, will now under the process of reduction will take place. So in reduction, the enzyme which will be used is beta keto acyl reductase, right? So it's gonna reduce. That means it's gonna add hydrogen, right? Now, as I told you in our first thing, if you remember over here, that whenever I talk about reduction, NADPH will get reduced to NADP plus, right? So this hydrogen will get donated, and over here also we have the same thing. So let us just try to see what is happening. So where did I reach? Yes. So NADPH will get converted into NADP plus, and then it will be reducing this particular ketone group. So whenever a ketone group is reduced, it will turn itself into a alcohol. Okay. Now, if you see properly over here, what do you see? Do you think that the ketone has been converted into alcohol? Yes, right? It has been converted into alcohol, right? So now this particular compound will be known as beta hydroxy ACP, right? Or we can also say 2 hydroxy ACP or 3 hydroxy ACP, and this is an alcohol, right? So now after the alcohol has been made, it will undergo a dehydration reaction. So the enzyme which will be responsible for actually removing a water molecule is beta hydroxyl ACP dehydratase. So dehydration will be taking place, that means removal of one molecule of water, right? So when a molecule of water is being removed, as you can see over here, this water will be removed as H2O, and then when this OH and one H will be removed, because this is an H, right? So now there is a possibility that the second and third carbon will form a double bond between them. So as you can see over here, that there is a double bond in between, right? And this particular compound is known as trans delta 2 enol ACP, right? So now why is it trans? Because the CH3 is gonna go out here, and then H is gonna go out here, right? So it's gonna be a trans. And then delta 2, because the second carbon has a double bond present in the second carbon, and enoyl because it is a double bond, and then ACP because it is attached to the ACP part of the FAS, right? Now, this is actually the result of the dehydration reaction, right? Now, after the dehydration reaction, finally there will be again a reduction. So it was CRDR, right? C, condensation, reduction, dehydration, again reduction, right? So NADPH will again be reduced to NADP plus, and then the enzyme which will be used is enoyl ACP reductase. So you just have to know, you just have to know these names, and then you'll be able to grasp the name of the enzyme as well, right? So now if you see over here, finally what will happen is this double bond will be converted into a single bond. That means hydrogen will be donated, and then when hydrogen will be donated, the double bond will shift to single bond, and this will be known as butyryl, right? So this compound is known as butyryl ACP. Okay, so as you can see over here that the ACP is attached to a ketone, and then three carbon molecules. So 4 carbon together, that's why butyryl. Now this is one cycle that we have decided right now. So if I just have to show you, where was the condensation? Okay, so this was the condensation reaction, right? This was the condensation reaction where everything was coming together. That means the acyl group in the cysteine was coming together with the malonyl group, right? And then they were forming a compound something like this. And then we have the ketone group being reduced to an alcohol. Then after alcohol we have produced a double bonded, right? And then we have finally produced a single bond, right? So we understood that CRDR is the process which helps us to make a straight chain. Now you must be wondering, is this all? Like, this doesn't look like a fatty acid, because a fatty acid looks like a long chain of carbon, right, along with a carboxylic group, right? So it's a very long chain. Now this has only 4 carbons, and the most common carbon that is actually produced is actually a 16 carbon compound, which is a palmitic acid, right? So a palmitic acid is a 16 carbon compound, which will be made. So I'm going to show you, now the other thing. So the one we learned was only one cycle. Now we're going to learn about a new cycle, right? So I mean to say, like, not a new cycle, but we're just going to keep on adding malonyl group, and then we'll extend the chain. So if you remember, in the first place I told you that malonyl coenzyme A is actually a building blocks of Fa, right? So now you'll understand why it is known as the building blocks of fatty acids. So let us just go over there. Okay, so I've just made it very simple over here. So as you can see over here, that the acyl malonyl group which was present, this will be transferred to the cysteine SH. So this group over here will be transferred to the cysteine part, right? And then the ACP group will now be free, and then it will be free for the new malonyl to come in. So now what will happen is, malonyl coenzyme A will come in, right? And then it is gonna pop off the coenzyme A over there, right? And then now as you can see over here, that the malonyl coenzyme A has successfully attached itself to the ACP group, and then the butyryl group which we had actually extended, it went to the cysteine group, right? So now what will happen? Now the same enzyme will come and then condense these two, right? So that was acyl malonyl transacylase, right? So transacylase, condensing, malonyl, um, acyl malonyl condensing transacylase condensing enzyme, right? So it's gonna come now down. So I'm just gonna show you how does it look like now. So I'm just gonna take the same. So we have, let me just show you, we have the carbon, we have the oxygen, we have C double bond O, right? And then we have two hydrogen which will be attached over here. And then as I told you that this particular part will pop off, it will cleave itself, right? So I'm not going to add this part, I'm going to rub it. So I'm going to rub this part. This is going to actually cleave itself. Just let me show you. Now let me add this part. So it has the C double bond O, right? It has the C double bond O and then C, right? And then we have in the pink, and then we have CH3. So as you can see over here, what did I do? I just I just made a new long chain, right? So now for when we started first, it was only two molecules, right? We started with two molecules, then we added malonyl, and then malonyl gave one carbon, and then we had four, right? Now we should have six. Now let us just check. 1 2 3 4 5 6. So 6 carbon. So now the same thing will repeat itself. That means this particular carbon group will be converted into a hydroxyl group, right? And then after hydroxyl group, it's going to convert itself into a double bond enoyl group, and then after enoyl group, it's going to convert itself into a single bond, that is a butyryl, right? Sorry, not butyryl, it's be like 6 carbon compound. Now let us just try to see. So after the formation of 16 carbon compound, as you can see, as we already did till here, right? So every time malonyl is gonna come and it's gonna add 2 carbon each. So 2 carbon it will be added, and then it's gonna form a 16 carbon chain. So now this 16 carbon chain is known as the palmitic acid. As I told you, it is one of the most common fatty acid which is synthesized. Now we need to cleave this off, right? We need to cleave this part off, and then we have to make this particular long chain of carbons into a fatty acid. But who is going to do it? So there is an enzyme called thioesterase which is going to cleave off the long carbon chain from the ACP group and then add an OH group finally, and then to this particular thing, and then we're going to have over here C double bond O, then we'll have an OH over here, right? And this is going to be the palmitic acid. Now palmitic acid is a saturated form. That means it does not have any double bonds. It's just going to have like the carbon between every carbon, like over here we do not have any double bonds, right? So this is a saturated carbon. Now let us try to see some other concepts related to this particular fatty acid synthesis. So one cycle, as you know, that we had a carbon dioxide being popped off, right? So whenever a malonyl-CoA was coming and then condensation reaction was taking place, a carbon group was getting out, right? And then in the dehydration reaction, we had one hydrogen coming out, and then we had been using 2 NADPH. So this is actually whenever you have to calculate something, that means in one cycle, if these molecules are being used, then in how many cycles, how many molecules are being used. So we must know how to calculate it as well. So as you know that in one cycle we used one malonyl-CoA, and one acetyl-CoA, right? And then we formed 4 carbon compound, right? And then as I told you that in each new cycle, a new malonyl CoA will be added. No acetyl CoA will be added, only malonyl will be added. Only malonyl will be added continuously until and unless there is a 16 carbon compound. And definitely, like, not only 16, there will be other carbon, there will be other fatty acids which will be synthesized as well, but that is like one of the most common one. So now let us try to see number of cycle will depend on number of carbons. That means how many cycle will a particular fatty acid synthesis will take place. So the in terms of palmitic acid, there is 16 carbon, right? So the formula will be n by 2 minus 1, which will be n is equals to number of carbons. So as we can say that if n is equals to 16, then how much it will be? This will be 8, right? So this will be specifically 7 cycles, right? So 7 cycles. So if you okay, if I just have to count over here, this was the first cycle, right? 1, then we'll have 2, 3, 4, 5, 6, and then 7, right? So 7 cycles will be there, because in the first one we made 4 carbons together. We were not synthesizing 2 carbons. First cycle was 4 carbon, then we were consequently adding 2 carbons each, right? Now let us try to just, you can try this at your home as your homework or something, where I have given you the name of the fatty acid, that is lauric acid, which has a carbon number of 6, and then we have a myristic acid which has a carbon number of 14, and then stearic acid which has a carbon number of 18. So you just have to divide, and then you have to just minus it, right? So you'll get the number of cycle. So the most important thing that you have to remember is actually the condensation, this part: condensation, reduction, dehydration, and reduction. And then you have to know the structure of malonyl and then acetyl coenzyme A. That will help you to understand these topics. And yes, like you must be finding it overwhelming, or you must be finding it very hard to understand for the first time. It's okay, you can this is something that needs constant revision. And you have, if you feel confused, you can always come back and then always again try writing down your copy, so that you don't forget the names of these molecules, and then you don't forget the process as well. So this is all about fatty acid synthesis. I hope you liked this video. If you did, please like, share, and subscribe. Thank you so much for watching. Hi everyone. In